Hi. Pleasure to be here. Thank you for inviting me. So I will talk about genomics, I will talk about the next generation phenotyping, and I will describe how we use the technology to help geneticists in clinic, research, and in the lab. So a little bit about myself. I'm CTO at FDNA. FDNA is a startup company, and most of what we hear about today is what we're doing at FDNA. My background, as Robin presented me, computer vision and machine learning research. And I work for the last 6 years to apply my knowledge in the field of genomics. I will start by a small motivation with a story about Morgan. Morgan is a 5-year-old girl from New York. When she was born, she seemed perfectly normal. As you can see, there is no sign for any problem that she might have. Her mother had a perfectly normal pregnancy, and there were no issues with her birth. So the first few months, everything seems perfectly normal. But as Morgan started to develop, few signs showed parents that there is some problem. She wasn't developing like other kids. She had problems to eat, problems to grow, problems to talk, to interact with the environment. Her parents started to look for answers, and they went to a lot of physicians. They took a lot of different tests, but the problem is that no test revealed nothing. It seemed like everything is okay, but Morgan is not developing the way she should. When she got older, she had problems to walk, to scroll, to get dressed, or even get sleep and wake up in the morning. It took 3 years to find an answer. And the answer was, after they took a test called whole exome sequencing, it found a tiny error in one of her 20,000 genes called NHRNPH2. Morgan was one of a handful of people, actually she was number 3, ever diagnosed with this variant. So there's barely knowledge in the world about this specific genetic variation. And on one hand, there is nothing to do because it's genetic. On the other hand, it helped her parents to understand what is the problem, what they should expect, to find similar patients, and to think together what they should do. This family even started a foundation to find more of similar patients, and they partnered with us to find such patients. Today, they already have more than 20 patients, and they try to influence pharma to develop drugs for their specific problem. So this story is a little bit typical story, and unfortunately, Morgan is not alone. There are more than 350 million people worldwide suffer from rare diseases. More than 7,000 diseases have been defined, and 80% of those are genetic. And every person with genetic disease have to go through a very difficult diagnosis process. Imagine that most of those people are children. They have to go through this diagnostic odyssey, which is really, really hard, and the literature talks about 6 years to get into diagnosis. So imagine, it takes 6 years to diagnose a patient. When we look on the diagnosis process, one of the main parts, and a very important part, is the clinical setting, where the clinician sits in front of the patient and need to identify different patterns, different associations to diseases, and find some direction for the disease, for the problems that the clinician sees. Those clinical signs called phenotype. So the clinicians have to assess the relevant phenotype to a set of diseases that might be relevant for this specific patient. Just to have some idea about what is a phenotype from Wikipedia. So phenotype is an observable characteristic of an individual resulting from the interaction of its genotype with the environment. So basically, the color of our eyes, the color of our hair, the appearance of the face, everything is phenotype. And it's very, very important for the clinician to be able to understand the different types of phenotypes in order to associate them with diseases. Now let's look on those patients and their phenotype. As you can see, there is something very similar between the patients in each side of the screen. On one side, we see patients with Down syndrome, which is a very common, unfortunately, disease. And it's very easy for us to understand that all those patients have similar disease and to identify that it's Down syndrome. On the other hand, we see Cornelia de Lange syndrome. Also, genetic disease, very rare, but you can see the pattern. The pattern is very clear. You could identify those patients pretty easily. So those patients have similar phenotype. And when we approach clinical geneticists and we discuss about their workflow, they tell us about how they take pictures of patients, how they advise each other based on pictures, how visual signs are very, very relevant for clinical diagnosis. So we thought, okay, how we can help this population with technology. And at FDNA, we developed an application called Face2Gene. Face2Gene is an application that is available on the cloud. It has clients for mobile, iOS and Android, and also on the web. And it's meant to serve the clinician in the lab. In general, the main flow is you take an image of a patient, and within less than 5 seconds, you get relevant diseases that are associated to these patients. We launched this application a few years ago, and it's currently used very widely across the globe. More than 70 clinical geneticists worldwide use us in 2,000 clinical sites and 130 countries. So it's spread very nicely across the globe, and we see good traction from clinicians. The flow of the application starts with taking an image of the patient. We see here Dr. Karen Grip takes a photo of the patient with her iPhone. And we build this in a way that it will be accessible for everyone. So it doesn't require any 3D imaging. It doesn't require any specific setting or no illumination, nothing of this sort. We simply went to the iPhone, to the mobile devices. And it's complicated in terms of technology because you have to address all those variations. But on the other hand, it allowed the application to be accessible to everyone. So Karen Grip is a professor from the United States, and she used it. And also, people in India with very, very limited resources use this application in the same manner. So once they took an image, they receive almost immediately, less than 5 seconds, results that show them correlation to different diseases. We can see here that they have some bars that indicate the amount of relevancy. And also, if they press on the syndrome, they get relevant information that teaches them about those diseases. Usually, those diseases are rare, and if a geneticist haven't encountered those in the past, they are limited with their knowledge. So this application also allows them to share knowledge. Now, we move to the more technological part. So the way it works, we have two main channels. One is the image channel, and the second one is the textual channel, which is basically user select information. We rely on a few databases that allow us to correlate between different phenotypes and syndromes and genes. And we analyze separately the faces, we analyze separately the text, and then we aggregate them together into one suggestions engine that allow us to present them with suggested syndromes, genes, and features to see what's relevant to the patient. It's a reference tool, and it also works in a refinement mode where they look on the suggestions, they can select what's relevant for a patient, and the system will recalculate the results, giving them more accurate results now, until they converge to some consensus between the machine and the clinician. Now let's look deeper into the facial part. So the facial part, we call it DeepGestalt, and it's basically a specific algorithm that we published recently, and we'll get into more details about that. The main flow is to take the patient's photo through predictive artificial intelligence, which is deep learning, as you can guess, and suggest syndromes that are relevant to this patient. Some challenges. The main challenge in this aspect is the database. So if we look on one of the most closest fields, which is face recognition, you can get a lot of images. You can just download millions of images from web of celebrities, and you have no problem. But in our setting, we have a big problem with that. We have small databases because it's really hard to find those patients. There is a large variation between the patients in different classes, between different syndromes, because in some syndrome, we have a lot of patients diagnosed. In others, we have only a handful of people. There is an ethnic diversity, which is also a big problem for us, because Chinese population with Down syndrome, they have Down syndrome, and also Caucasians have Down syndrome, so they are in the same class, but they look differently. And we need to support a lot of classes. Basically, there are 2,500 diseases known today to our research with facial patterns. So we need to be able to support all of those syndromes eventually. Some on the comparison for face recognition. In face recognition, it's a very challenging task, which deep learning helped a lot to get good results on, and they have a lot of variation. One variation is the age variation. Another variation is occlusions, illumination, different appearance of the face. Sometimes you smile, sometimes you cry. So all those are challenging for face recognition, which we call intraperson. But in our case, we also have the intra-syndrome variation, where you actually have to overcome the identity. In face recognition, you preserve the identity. In our case, you preserve the syndrome, and you have to be robust to the identity. So all those patients you can see in each one of the rows are different people with the same syndrome. So the algorithm has to learn only the patterns that are relevant for the syndrome. So it makes it more challenging for us. The way DeepGestalt works, this is a publication we published in Nature Medicine a few months ago. It takes the image through a deep convolutional neural network and produces a probability for a list of syndromes. We currently support more than 300 syndromes in this architecture, and it grows as we collect more data and are able to serve more patients. Some words about how we train the system. Basically, we have two steps. The initial training, where we take the architecture, we took specifically an architecture that has relatively low amount of parameters, so we haven't gone with the full-blown image recognition architectures, which are, they have to have like millions of examples in order to boost. So we took something that is relatively less consumed with parameters, and we train it first for the face recognition task. So we collected images of celebrities, as I said before, and we were able to teach the network what is a face. What is a face? What is the statistics of the face? And each layer learned about the statistic. Eventually, we got good results. We compared them to the common benchmarks, and we saw it works well. Then we use transfer learning. Transfer learning is a technique in deep learning that allows you to take a pre-trained network and to transform it to a different domain. And in our case, it's very useful because we were able to take a small amount of images. It's relatively small database. It has a few thousands of patients, but still it's slow for deep learning. And once the network already knows what is the face, how the face looks like, it was easier for it to learn to identify syndromes. So I think about it as teaching a child to recognize a new car while he already knows what is a car. So it's easy for him to learn. So this worked pretty well for us, and we're able to provide good results. In our paper, we show some of those results. So the first top results are about binary comparison of our technology with Cornelia de Lange syndrome, which we saw an example before, and another one called Angelman. And you can see that in the bottom of the table, our results versus the results that were published before, and those results are the comparison to humans. The techniques that were used before and the previous papers talks about how well clinical geneticists can identify those syndromes. And you can see, for example, that they were able to achieve 87% on this benchmark, and we 96. And so this algorithm was able to overcome human performance, which is nice. Then we expanded it to multi-class Gestalt model, and that means that we support a lot of syndromes at once, 300 syndromes, as I said, and we also achieve very good results. There is a problem in this field with benchmarks because it's hard to compare to when you present new technology. So what we did, we also released some benchmark to the public so that further research can be done, and the field will hopefully will move forward. Another part that we showed is that this algorithm can also identify different genes, not only syndromes, but genes. And this is a change in paradigm. We'll see it in the next slides. The other part of our algorithms is the textual part, where the user selects information, facial features or other features in the patient, and we treat them as selected textual description, and we also try to reason about the suggested syndromes. So this is done in more statistical analysis, where there are two actual relevant information parts. One, all the facial features and all the features in general in human can be described in a hierarchical way. So there is a database called Human Ontology, and this database allows us to move through the ontology and associate the known probability of each one of the features to syndromes and distribute it across all the ontology. So this one aspect of our algorithm, and the second one is TF-IDF. It's a very known algorithm to retrieve information. So we look on syndrome as a document, and we try to retrieve the information based on the query that was submitted by the user. The combination of the two provides pretty good results. Another aspect of textual analysis is clinical notes. Clinical notes are well used in this field. The physicians, they type what they think about the patient, and they submit it to the EMR. And we built an algorithm that is able to analyze those and extract relevant features from those clinical notes. So we're also currently in a process of publishing this work, so it shows that we have nice results. Another part, which is very important for our application, when you approach clinicians and you say, hey, I have an AI system for you, start using it, they don't believe you. You have to convince them somehow that it's real, that it works. So we decided to provide them some visualizations that allow them to understand what the algorithm thinks about. One of them is a heat map. In this heat map, they can see a correlation between the facial appearance and the specific syndrome. And as you can see here, some areas are more red than others. And hopefully, if we do it right, they can see the facial features that they think that are relevant to the syndrome in this picture and say, hey, okay, I get the intuition, I understand it. The way we do it, we simply use backpropagation. So the method of backpropagation is that you can take the syndrome that you want to examine and its probability, and to feed back the information to the pixel level. And then you get a heat map, which is not well understood. But if you have a prior knowledge on the face and we know where each region resides, then we can provide this heat map that allows us to show the relevancy for different facial patterns. Another part on instant delivery, I'm getting back to the slide of our patients that we saw before. And we can see this visualization. It's a simple average of all the faces into one image. And as they look like a real patient, there is no patient that looks like that. It's an average. And it's also, we found that this simple visualization helps our audience to understand how would a computer see this syndrome. Like when you approach the clinician and say, hey, we built a model that is able to classify the syndrome, it's hard for them to grasp this idea. But when you say the model might look like this, they understand it. So to conclude this part, in Phase2Gene, we created 3 pillars. There is a network, which is all the network of our users. There is a data. When they use our applications, they provide data, and they provide feedback, and they allow us to build more technology and improve our technology, which in turn we put in our application, and they can use it, and this cycle feeds itself. So it was very useful for us to get data to be able to improve the results as we go. Another part that allowed us to improve the results is the part that deals with research. So we found that our users are very prone to research because it's a field where they always strive to find new genes, they always strive to find new patients. So we created this application called Research Platform aside from Phase2Gene, and they can simply go there and check their hypothesis. If a doctor has some hypothesis, they can upload cohorts, and they can run our algorithms on top of those cohorts. So it's similar to the transfer learning that I showed before, but it's an automation. They can do it by their own. They don't need us to hold their hands. And this allows our users to publish a lot of publications. In the recent year or so, they published more than 20 publications. And each one of those allowed us to discover something new about this field. And for us to get some data, to understand better where we should go into more depth. And one of the interesting things about it is that those clinicians, they usually have data silos. So a clinical site, they work for 40 years, and they have a lot of data reside in their boxes or in some laptop or whatever. And we found that this research activity is a good motivation for them to take this data, to analyze it, and see what they can understand, what the new generation can learn from this data. And it actually created value for everyone because they were able to publish new research, they were able to get their data to be used by others, which is something people usually want. And the community had the benefit of having this knowledge shared with everyone. Some examples, so there was a group that looked for the ethnic diversity, and they were able to show us that even for Down syndrome, that it ran on Congolese population, we were pretty bad in results, and we worked together, we improved that, and the publication that published showed that the results were pretty good. Another part is the age diversity. So there was a group that took a specific syndrome, and they analyzed different aspects of this syndrome versus age, and they found that it's pretty robust, that it works pretty well, which is also nice because it also allowed the other users, when they see this, to trust our system and better know when to use it. Another interesting part is the association between the genotype and the phenotype directly, and this research showed that it's feasible to not only classify for syndromes, but go straight to genes. And it's not trivial because essentially the phenotype is translated to syndromes. Syndromes are diseases. But genes is something that is probable. It's not necessarily correlated specifically to a pattern that you can see. So this research showed that it's possible to do that, and it was a good motivation for our next part. The next part talks about precision medicine. So precision medicine, it's something that people talk a lot in healthcare these days, and it's considered to be the future of healthcare. And it all starts with genetic testing. Genetic testing recently is called NGS, Next Generation Sequencing, where your DNA is being sequenced, and they can see all our genes. So it sounds great. Someone knows everything about my genes, and people imagine that it will provide a lot of new tools, which called precision medicine. So the tools will be fitted for our needs, for a specific patient, some services that are relevant for me only. They know everything about me. It's all about a specific patient. So this technology called next generation sequencing, and it's meant to change healthcare. But it's not so trivial. And it seemed like there is a process where in a good shape in this process, but it's not yet adopted by the consumers. The industry talks about next-generation sequencing being currently in a clinical adoption phase, which is one step after research, so it's good, can be used in clinic. And indeed, we see that in recent years, the cost of NGS has dropped significantly. The more genes are being sequenced, and more knowledge is accumulated about those activities, and in turn, they're able to diagnose more and more syndromes. So the trajectory is very good, but it's still not in the consumer part. It's something that evolves. It turns out that it's very hard to do NGS, and the main problem is data. So the highest barrier for the adoption of NGS is the data interpretation. So it turns out that when you sequence a person, you get all the person's genes, and you have like a book with all the genes. And now you have to understand which one of those genes causing a specific problem. So what you do, you compare to the normal population, and you compare to some models, and you try to understand which one of the variants are causing some problem and which ones are not, and those that causing a problem called pathogenic. So a variant is basically a difference between my genes and the model, the regular population. So well different, right? So we have a lot of variation between each one of us and the normal population. So when you sequence a person, you end up with huge amounts of variations, and then you need to analyze them. So bioinformatics today, they know how to analyze most of those variants, and they just filter them out. But eventually, they end up with still a big amount of variants, sometimes thousands of variants, which they need to go one by one and to see if this specific variant is relevant for a specific problem that the patient has. And it actually works like that. In the lab today, people go one by one, thousands of genes, and try to understand if this gene might be the pathogenic causing gene. So it's kind of hard, and they talk about finding the right variant is like finding a needle in a haystack. The community also knows that phenotype helps this process a lot. Eventually, all the genes, all the variants, it's a probability. Some might appear in the patient, some might not. If my parents have green eyes, I have the gene. I might have green eyes, I might not. There is a probability for that. But in order to know specifically if I have this green eyes, someone needs to look at me and my eyes and to see it, which is looking at the phenotype. So the industry today, they use the phenotype, but the problem is that their tools are very old, and the amount of information that they're able to use is very, very small. Essentially, they use very, very high-level descriptions. Like, for example, this patient has intellectual disability. Well, a lot of genetic diseases have an intellectual disability, and it doesn't help most of the times to know that. So we suggest a new pipeline where our facial analysis is integrated into the variant analysis process, and together it's supposed to boost the results. This is the idea, and this idea started with one of the research that I described before, about how you reason from patient's photo to genes directly. And we've worked on this for a couple of years now. And actually 2 days ago, this paper was published, which is a collaboration between our company and Bonn University, led by Professor Peter Kravitz. It's called pedia. It's a prioritization of exome data by image analysis. And basically, we took this idea of adding the facial analysis into the pipeline of genetic interpretation, and we built a study around that. We had a call for participation. We took a lot of patients, and we were able to prove that this is actually helpful. So we go into some details of this method. So the main idea is about taking the patient card. So we're not looking only on the face. We're looking on the patient card, everything we know about the patient, and we see it as two groups. One is the photo, and one is the textual information. The photo is analyzed by DeepGestalt, as we saw before, and provide a score for each one of the genes now. We took the scores that we provide for syndromes, and we transferred them to gene through some databases that allow us to do this association. Then we use the textual analysis to do the same thing, to provide now scores for each one of the genes instead of syndromes. And we also added some other algorithms to boost the results called Phenomizer and BOCA, which are most used by others groups as well. The genetic part, which is actually the variants that were sequenced from the DNA, are fed into an algorithm and provides a CAD score. CAD score is a score that predicts the pathogenicity of each one of the genes, how likely this gene to be a pathogenic one. And then we took all this information and we feed it through another machine learning step, which uses a simple SVM. And basically what it was able to do is to take all the scores and to provide one single score from those scores by a linear simple SVM. And when we look on the distribution of those scores, we can see in part C here the Manhattan plot. Manhattan plot is a plot where you, on your x-axis, you have the genes in the way they're in their chromosomal position, and on y-axis, the score that this pedia algorithm provided. And hopefully, we get to see these nice results when one gene is upper than the others, and that means that this gene has the biggest probability to be the pathogenic gene for this specific patient. We checked this approach on the benchmark. It's a relatively big benchmark of almost 700 patients. And we've seen really good results. So usually in this field, we look on top 1 accuracy and top 10 accuracy. And when we look on top 1 accuracy, you can see a boost from 16 to 90% in having with only genotype or genotype with our pedia approach. The middle one is having the legacy phenotypic information, which is without the facial analysis. So the results have improved a lot. Also for top 10 accuracy, we also see a significant boost. And with our new approach, we achieve 98%. So if we get deeper into what it gives, we can reason about the impact of each one of those algorithms. And we can see on x-axis the rank of the syndrome, which means how many genes we need to analyze, and on the y-axis the sensitivity. And this curve allows us to reason about which one of the algorithms is more impactful for this analysis. We also applied it into another research in a different site, which also showed similar results. So the red one here is the pedia approach versus other approaches where the blue is a legacy approach in Greenwood Genetic Center in the United States, where we actually compare their real-life work. They have some system which allows them to include the phenotype, but it's limited, as I described. And this is a comparison versus their system. It's not like a research setting, it's a real-life application. And we see here a really nice boost in results. So this is just a recent study from a few days ago, and we see here how much is approved. This whole approach also can be used in another way for engineering. So if I am a lab director, I need to somehow balance between the time I invest into the analysis of those variants and the performance that I might get. And we look on this ROC curve as a way to do that. So once we have the pedia score, it's a single score that captures all this information. We can now reason about the performance and the time that invested. So if we look on the curve, on x-axis is the time, so it's also correlated to the amount of genes that we sequence, that we review. And y-axis is the diagnostic yield, the performance that we get. So everyone knows that if we add the phenotype, we increase the performance. But now we also shown that if we add the images, we also take the performance closer to reduce the amount of time that they can invest. And when we look on the pedia, we can actually somehow tune it. We know now that if we sequence top 10 results, we get a certain amount of genes that we need to look at, while we know the performance that we get. If we, in a different lab, will take more genes, it will boost the performance, but we'll need to invest more time. So this also is an engineering tuning way for the lab directors to tune their work. And for a machine learning guy, it's obvious. We do it all the time. But for the lab directors, it's not obvious. They're not used to work like that. So it's a different paradigm for them. I'll now conclude all the talk in a general review about FDNA is our company, and basically we deal with 4 different fields. One is the clinical support, where we provide tools for clinicians. As we saw, the clinical site, taking an image, getting results, improving your workflow. The second one is the genetic interpretation, the one we just saw with pedia, the ability to improve the lab work, improve the genetic interpretation by incorporating our algorithms into the workflow. The third one is biomarker discover. The research applications that I described allow us to find new patterns, new associations, and promote the field in a research aspect. And the last one I haven't covered, but I'll tell about it in 2 words. It's a patient discovery. It turns out that with all our tools, we have more connection to patients than pharma. And pharma, when they try to develop some drugs and they try to do clinical research, they look for those patients. And they look for patients with specific phenotypic characterization. And we are able to help them. So we now partner with some pharma companies to help them find more patients and hopefully reduce the time from 6 years of diagnosis into much less than that. The future for us in terms of technology is to develop more the phenotypic coverage. So we're currently able to support the facial and textual parts. We're also investing into X-ray of hands, mainly brain MRI, some other signals such as video and voice also explored. And as we go along, we'll have more coverage. We'll be able to reason about more phenotype, and we're able to boost this field even further. Thank you.
Thank you, Yaron, for this very inspiring talk and impressive results. We have time for a few questions.
Hello. Thank you first for your really inspiring talk. I have one question because you mostly have shown images of young patients. And do you specialize on younger patients, or does your algorithm make a difference in the disease recognition if the patient is very young or if he's an adult?
Okay, it's a good question. So it depends on the syndrome and the onset of the syndrome. Some syndromes, they start to develop in a very early age. And for our application, it's most relevant to focus on children. Because if you're 30 years old and you have some disease, you probably know about it already. And by the way, it's not always true, but in terms of the majority of the people, you better focus on children. But we also had some experience of testing this approach on older population, and it works, but it requires tuning, because the facial appearance of children is different from the grown-ups.
Hello, a very inspiring talk. I was actually wondering how important motion, facial motion would be in the analysis. I would assume it gives you a big additional differentiator. So right now you only analyze static images, is that correct?
Yes, we currently analyze only static images. So motion, I can think about it in a more looking on the whole patient. If you look on the whole patient, you can reason about motion. And some diseases, we know that from some patterns that the patient does, you can see, you can reason about the condition. I'm not sure about the facial motion itself, but we do explore now video. So in a video of the patient, you can see all those signs.
More questions?
Thanks a lot, Yaron. I was quite surprised that the neural network is so small. Yeah, I had supposed that you had been using like ResNet 152 or so, and an enormous number of free parameters. Would you maybe explain to us a little bit more why it was so advantageous to have this relatively compact network?
Okay, so as I explained, the basic database of the facial images is relatively small. And when you look on the trajectory of face recognition, there was at some point where Facebook and Google, they had huge databases and they were able to train those big networks. And at some point, there was a Chinese group, I think, they published their own database. It was small, and they said with a small network, we're able to get to similar results. So the literature for face recognition also supports that. You can have a relatively small network and get to really, really good results. And it's nothing special. It's just the way the filters are organized, the way the filters are used. So if you stick together a 3x3 filter and another 3x3 filter, you get effectively a 5x5, but you have less parameters, and you try to use less fully connected layers, and you use more convolutional, then you also reduce parameters. So it's not so sophisticated layers, but you use them right, and you get eventually less parameters than the full-blown ImageNet. And it works well. I mean, when we explore other applications, other architectures, we get boost in performance, but it doesn't worth it, usually.
Do you reckon that in the future, privacy issues may arise from this kind of technology? Because that's quite personal information, whether I have such a syndrome or not, or what kind of gene variations I have. And if you can do such amazing things by just looking at a picture of somebody, do you think that maybe that could be used in some precarious ways sometime?
Maybe I have time to answer you. Okay, so it's a really, really good question. I'll answer in two aspects. So first of all, for us, the way we build the application, we really care about the privacy because no one will use it if it's not secure enough. So we keep all the standards, if it's a HIPAA, GDPR, ISO certification, whatever we need it. And we also build the algorithms in our research group in a way that they cannot look on the images. There is some part of the images that they can look at, some research data sets, things like that. But when the training is done, it's fully automated, and there is no human that interacts with the patient's photos. So that's on our technological part. But for your question about what people can do with that in the future, I think no one knows right now. But if you look also on the genetic side, in genetics, people think it's information that need to be protected, right? So in the United States, they have the GIS laws, and they somehow protect the genetic information because they think that they can reason about some diseases that you might have. But no one thinks about phenotypic information. And yes, we show that phenotypic information is also relevant for your diseases. So what can they do with your Facebook photos, with your Google photos? I don't know. That's a question we need to think about. It seemed like currently there is no regulation on that. But I think that the way we have regulation for genetics, we also need to think about phenotyping.
There recently have been some developments a few months ago, especially by Mozilla, to do a data set that has better support of different ethnicities for face detection and similar things, to remove biases from the data set. What approaches do you do so that your data set, because it's especially small, you make sure that there is less bias on features that you don't want to have a bias on?
Yes, so we do have bias. It's inherent into the algorithm, the database, and the knowledge that generally clinicians have. Most of them came from Europe, and they biased by their nature. But we try to analyze different syndromes and to see where we need to boost the performance. So for example, the study that we shown on Down syndrome, we saw significant bias for Caucasians versus other populations. And then we added images intentionally of different ethnic groups, and it improved the results. We also found, we also did it for other ethnicities, Chinese population and different syndromes, and we found also interesting thing that it's much better for the network to have everything in one model, and it's able to generalize better rather than split it into classifiers that are specialized on different ethnicities. So it's also something that we learned along the way.
So maybe one question from my side. How easy is it to transfer your results to other medical fields? So you also mentioned that you do text analyses on the notes of the doctors, which could be applied to like any general medical field where symptoms are written down and you can aggregate them to get to a diagnosis. So how easy it is to transfer this to other medical fields?
So from my experience with machine learning and deep learning, on very high level, it all seem very similar. But the devil is in the details. So I think that one needs to better understand the database, better understand the specific field, and only then you can answer this question. In general, you can take the ideas, you can probably take the code. I don't know if it will be 80-20. I don't know if it will, if you cannot improve it more. I think it could be a good baseline for every other field, but eventually you have to deal with your data and specifics of the problem.
Hey, so adding on to that question, I've seen that a lot of clinicians and a lot of doctors also just write notes rather than digitizing there and typing it on a computer. So do you also take that data, or do you take like actual notes from doctors and feed that in the network as well, or is it actually all just digital data? And going back to that, doctors have their own short forms for things, and like certain terms, they just write 3-letter acronyms. Is that all recognized at the same time?
Yes, so clinical notes is a field that we're trying to expand. Usually, they do have a specific way to write clinical notes. Some doctors write a lot of acronyms. You're right, it's hard for the algorithm. We try to find ways to overcome this. Usually for us, it's relatively focused because we try to look for the ontology descriptors. And if we look for a long distance between the eyes, there is few ways to say that. And we try to learn all the possible ways by looking on the notes. So yes, we need to analyze them. We have a lot of back and forth with errors. And it's a field that we're developing currently.
So you have been focusing on the identification of diseases to pinpoint phenotype to a genetic disease, but of course you could also use it for the monitoring of diseases. So we have many genetic predispositions which have a, let's say, a lifetime penetrance of only 5%. And you could monitor a population over time to identify whether a certain predisposition is actually manifesting. Is that an idea you are following?
Yes, so we explored during the years some applications of taking pictures of, or asking parents to submit pictures and then alerting something that happens or some phenotype that starts developing. I think we're not there yet in terms of the community, the technology, and the adoption. But yes, it might get to the point where you're sequenced once, your healthcare provider knows your DNA, and then you have some probability for certain things, then you're tracked and the phenotype is monitored and then identified at the right time to give you the right treatment. So I think it will go there, but currently it's not there yet.
Okay, thank you very much again for this nice talk.
Thank you.